Okay, so this is the second time I'm recording this lecture for you guys. There was something happened with the camera first time I did it, so this is second try on my part. Sorry you won't have access to the questions that the students in the face-to-face class asked, but at least you'll have the content. So we left off talking about the cell membrane and the intercellular matrix. We need to talk a little bit about how stuff moves into and out of the cell. And there are really two ways in which that's accomplished. There is a passive process, or a group of passive processes, and an active process. And what differentiates passive and active is whether or not energy is expended to move that substance. So in passive movement, we don't have to expend any energy to move something from one side of a membrane to another, for example. And diffusion is a really good example of a passive process. With diffusion, what happens is, as a product of random or chance events, some substance moves from a high concentration to a low concentration. Now, that's exemplified here in this image where we have a flask of water, and into that flask of water, we put a couple drops of blue food coloring. And in the beginning, that food coloring is all clustered together. But gradually over time, as a product of the random movement of molecules, that substance diffuses across the water, so that it becomes ultimately kind of a ubiquitous kind of bluish color. But the important point here is it's going, the net movement, even though it's a product of random or chance events, the net movement is from a high to a low concentration. Okay, so membranes make things membranes are selectively permeable. What that means is membranes will just naturally allow some substances to diffuse across them, and other substances they are going to repel. And that's a product of the bipolarity, or the dual nature of the phospholipid molecule that makes up most of the plasma membrane. Remember, the phospholipid molecule has a hydrophilic head and a hydrophobic tail, and when clustered, those hydrophobic tails really stick together in that phospholipid bilayer. Because there's a hydrophobic core, anything that is polar or anything that carries a charge will not be allowed to diffuse freely across the membrane. So things that carry a charge, like ions or water that carries partial positive and negative charges, will be held out by the plasma membrane. However, things that don't carry a charge can pass freely, diffuse freely across the plasma membrane. So things like oxygen gas, carbon dioxide, and nitrogen gases can all diffuse freely across the plasma membrane. Now, with diffusion, there is this idea of a gradient, right? Things naturally move from a high to a low concentration. And the speed of movement is going to be determined by a few characteristics. That is, how fast something moves from a high concentration to a ubiquitous distribution. One of those things is the steepness of the gradient. The original concentration of the molecules will determine how quickly things diffuse. The steeper the gradient, that is, perhaps the higher the concentration of one substance on one side of the membrane as opposed to the other, will determine how quickly things move across the membrane. The higher the concentration, the steeper the gradient, and the faster things will move. Another important phenomenon or characteristic that determines how quickly passive movement will occur is the area of the membrane. So plasma, the phospholipid bilayer, is really what makes things selectively permeable to polar and nonpolar molecules, kind of. When we think about oxygen gas specifically, it can move freely across the plasma membrane because it doesn't carry a charge, so long as there are independent phospholipid molecules adjacent to each other, so it can slip in between adjacent phospholipid molecules. But when we start throwing other things into the plasma membrane, like protein and cholesterol, things like that, the amount of surface area occupied by the phospholipids decreases. So really, the higher the concentration of proteins in a certain area, the lower the rate of diffusion, because there's just less surface area for diffusion to occur. And then finally, physical characteristics like temperature and pressure and electrical gradients can also influence how quickly it is things move across the membrane. And that makes some sense too, right? Because this is a random process. It's a product of molecular movement. And if we increase or decrease the rate of molecular movement, it will either increase or decrease the rate of diffusion. So, charged or polar molecules, or ions, atoms, whatever, aren't the only things that we want to keep out or allow in. Now, we want to have some charged particles coming into the cell, right? Like water's a really good example of that. We need to have water moving in, but the hydrophobic core of the plasma membrane keeps it out. So for that purpose, we have carrier protein molecules. And these really exist in, well, a couple of formats. One is a protein channel, and one is called a carrier molecule. Now, protein channels are just like tubes or straws through the plasma membrane. They're open channels that allow anything that fits into that hole to pass through. We have protein channels called aquaporins, and they are specifically for water. And really, anything that approximates the size of water and the charge of water can move freely across these protein channels. Now, it's a passive process because water will always follow its concentration gradient. If there's a higher concentration of water outside the cell, then that water is going to want to flow into the cell. But if there's a higher concentration of water inside the cell, then it will naturally want to flow out of the cell. Alternatively, we can have carrier proteins, and these are selected in the kinds of things that they allow across the membrane. What happens with a carrier protein is there is an active site in the middle of that carrier that is selected for a specific molecule. And when that molecule, let's say that this protein is opened up on the other side, when that molecule binds to, when that protein binds to its target molecule, then the conformation or the shape of that protein changes and it opens up on the inside, and it allows that substance, but only that substance, through. Now, carrier proteins are selective in the kind of things that they'll allow in or take out, but they're not selective in directionality. That is, even if this is an important molecule for inside the cell, if there's a higher concentration of it inside the cell, this same protein will pick it up and allow it to follow its concentration gradient and dump it back outside the cell. That's called facilitated diffusion. This passive process is facilitated, made possible by this carrier protein. An important type of diffusion is osmosis. Now, osmosis is defined as the diffusion of water across a semi-permeable membrane. And there are a couple of terms that we need to be familiar with that will really dictate how it is water is moving across a membrane. The first term is solute, and that is anything that's dissolved in water. So in salt water, the solutes are sodium and chloride, right? Sodium and chlorine ions. Solutions can either be hypertonic, which means they have a very high concentration of solutes, which means that they'll want to pull water towards them across a semi-permeable membrane, or they can be hypotonic, which means they have very few solutes dissolved in them, which means it's going to want to push water across a semi-permeable membrane to try and equalize the concentration of water on both sides of that membrane. Or there could be isotonic solutions, where there really is no concentration gradient on either side of the membrane with respect to water. Or said perhaps a little bit differently, the water on both sides of the membrane have an equal amount of solutes dissolved in them. When solutes are dissolved in water and osmosis occurs, it's important for you to remember, however, that really what moves isn't the solute, it is the water. Osmotic pressure is defined as the amount of pressure required to stop the movement of water. So if there's a very high concentration of water on one side of the membrane and a very low concentration of water on the other side, it's going to be very high pressure. That water is going to want to move across that membrane very, very rapidly. But if the amount of dissolved solutes are about the same and the concentration of water is about the same, that pressure will be much reduced. There is another term called hydrostatic pressure that's also important for us. And that's defined as the pressure of water inside the cell that's pushing out against the plasma membrane. In animal cells, where we don't have a cell wall that's constraining cell shape, hydrostatic pressure becomes very important, because if we have too much pressure inside the cell, then it will actually cause the plasma membrane to break. It's called lysing. The cell gets lysed or cut open by water. So this figure shows you animal and plant cells in hyper, iso, and hypotonic solutions. And what you'll notice is that in hypertonic solutions, animal cells get shriveled. In isotonic solutions, they maintain the shape that they're supposed to. This happens to be a red blood cell. In hypotonic solutions, the cells take on water and they increase in size. Plant cells do the same thing. In a hypertonic solution, that cell is going to shrivel up. The plasma membrane is actually going to pull away from the cell wall. In an isotonic solution, the cell, water isn't moving either in or out of the cell, but that cell becomes somewhat flaccid. With a hypotonic solution, water is constantly going to be moving inside the cell and causing a hydrostatic pressure inside a plant cell. And that's an important thing for a plant cell. And we'll talk about why in just a second. So if we have a beaker of water, and in that beaker of water we insert a semi-permeable membrane, and we have an equal amount of water on each side, then that water is not going to move. If, however, we introduce a solute, in this case urea, which is a salty solution like urine, into the water, all of a sudden we've changed the concentration of water. So that water is going to want to move from the high concentration to the low concentration. By adding solutes per unit area, we decrease the concentration of water. And that's what we see here. The water level has actually risen in this one because we're trying, our bodies perhaps, or this beaker perhaps, is trying to equalize the concentration of water on either side of this semi-permeable membrane. Okay, so we're able to maintain osmotic pressures within our cells, and that's important because, like I said, if they take on too much water, they can break. If they take on too little water, they actually become shriveled up. And as we know in biological systems, the shape of things is important, it determines their function. There are some cells that have vacuoles that are actually able to pump water out of the cell. It's called extrusion of water. And we talked about those in the context of protists. So protists have contractile vacuoles that actively pump water out of the cell. Alternatively, we can bathe our cells in isosmotic solutions or isotonic solutions. We do that in our blood. The albumin of our blood is isosmotic, it's isotonic with the insides of the cells, so it allows the cells to maintain their shape. But plants want to maintain a positive pressure of water inside their cells. It's called Turner pressure. And when that water is pushing out against the cell wall, it allows the plant to be rigid in space. So think about a stalk of celery in your crisper drawer that you've left there for too long. If you pull it out, it's flaccid, it becomes somewhat limp. But if you were, because there's no positive Turner pressure flowing water into the cells and causing them to be really taut and holding that stalk upright. But if you put that stalk of celery into water, that water would naturally be drawn into the cells, and your stalk of celery would become turgid again. It would not turgid, I'm sorry, it would become solid again. It would become really stiff. Okay, so that is passive movement. And remember, the salient point with passive movement is that substances, whether it be a solute or whether it be water, is always following its concentration gradient, always moving from a high to a low concentration. But sometimes we want a higher concentration of something inside our cells than there naturally occurs outside our cells, like a higher concentration of sugar inside our cells than naturally occurs in the environment. Sometimes we want a lower concentration of things inside our cells than naturally occurs in the environment. So in that case, we're moving a substance against its concentration gradient. That is called active transport. And we actually need to expend some energy in the form of ATP to move a substance against its concentration gradient. So when we move something into the cell in bulk, that's called endocytosis. And we do that by creating a vesicle around that substance and pulling it into the cell. And you see that happening right here, where there is some substance out here outside the cell, but the cell is recognized as something interesting, creates this vesicle around it and pulls it inside the cell. Now, if we do that with some organic material, like some food particle or something, it's called phagocytosis, or cellular eating. We can also move water into the cell that way, and that's called penocytosis, or cellular drinking. And that's what you see down here. There are some water molecules that are collectively interesting to the cell, so it creates a vesicle around them and brings them into the cell. We can do the opposite. We can move things in the opposite direction too, and that's called exocytosis. And that's where we're creating something inside the cell, whether it be a waste product or some protein or cellular product that's needed elsewhere, and we move it in mass to the surface of the cell, and we just extrude it out. Sometimes, a particular type of endocytosis is called receptor-mediated endocytosis. And that occurs when a cell has a number of protein receptors on its surface. And once some number of those protein receptors have bound to the molecule they're most interested in, then a vesicle is created around those receptors and it's pulled inside the cell. A good example of this are LDLs and RBCs. So LDLs are low-density lipids, which form plaques inside our blood vessels. You build up fat, and it leads to heart attacks and strokes. Red blood cells, however, have receptors on the surface of their cells that identify LDLs. And when they identify them, and a certain number of them have been identified and bound to those receptors, then collectively they're brought inside the cell. Now there is a mutation of the gene that codes for those receptor proteins. And if you carry that mutation, then your body isn't able to identify LDLs in your blood. And if you're unable to identify those LDLs in your blood, then ultimately you get